This is Guadalupe Fernandez, and I work as product specialist at Cytognos. First of all, I would like to say thank you for your time today and for being here with us. In this session, I want to present our new solution for the identification of Tcell subsets. In this presentation, we are going to focus on the composition function and possible application for this antibody combination that I'm going to present you today. Let me to say thank you to Biospecific, our partner in Australia, New Zealand, and Hong Kong. And specifically, Steve, thank you for helping me to organize this session. Let me just to show a quick reminder about who we are and what we offer. Cytognos is a biotech company in the flow cytometry field over the last 25 years. And we are based on the Euroflow investigation. And specifically, we are providing solution in the oncomatology, but also in the clinical immunology field, but also in addition for the clinical trial performance. And in this presentation, we are going to present a solution that is applicable and useful in these three areas. And this solution is designed to ensure that in the lab, we are going to obtain and to enhance the patient care, but also we are going to improve the laboratory efficiency. So these are the points I will go over today. First, I will start giving you a quick review about the immunophenotyping of Tcells, and then we will cover some interesting advanced software tools, and we will review several clinical applications for the antibody combination that we are going to see today. On the right part, you have a chat box, so feel free to write any question you may have, and at the end, we will have some time for questions. So let's start with a quick overview of the memory generation process in Tcells. In homeostasis, naive Tcells that are egressed from thymus recirculate between blood and secondary lymphoid organs, such as the spleen or the lymph nodes. But upon recognition of their cognate antigen presented by the antigen-presenting cell, the naive Tcells get activated and differentiate into effector Tcells, which are going to migrate to peripheral or inflamed tissues, and they are going to collaborate with other immune cells until the antigen is cleared. After clearing of this antigen, most of the effector cells undergo apoptosis. However, a small pool of Tcells turn into long-lived memory Tcells. In peripheral blood, there are two main types of circulating memory Tcells. These are the central memory Tcell and the effector memory Tcell. The central memory are characterized by a high potential of cell renewal and the expression of molecules that are implicated in the homing to secondary lymphoid organs. And however, this central memory Tcell lack immediate effector function. So, in the contrary, the effector memory Tcells produce effector cytokines such as the interferon, the AL4, AL7, and express protein involved in the migration to peripheral tissues. In this graph, you can see one of the most accepted model. One of the most accepted model of transition from naive to memory terminally differentiated Tcells. The model proposed that quiescent naive or memory Tcells turn into activated effector cells and progress through the maturation pathway. But this maturation pathway is not always linear, and cells can jump into different stages. So during this transition process, cells are going to lose stemness and proliferative potential and lympho homing capacity, and they progressively become more cytotoxic and produce higher amount of effector cytokines. And at the same time, the cell also gain tropicins to inflame tissues, and also they gain senescence marker. So after this introduction, we can move directly into the solution that to see how we can analyze the different T-cell subpopulations. So this is a 7-color panel for the identification of the different subpopulation of naive, memory T-cell, and effector T-cells. We have included CD45, CD3, CD4, and CD8 for the these markers are used to identify helper and cytotoxic T-cell population. And besides, we have include additional marker to classify the CD4 positive and the CD8 positive T-cells into the different subpopulation of naive and memory T-cells. So we have included the CD27. This is a co-stimulatory molecule whose expression is lost in effector T-cells. We have also included the CD45RO, which is expressed in memory T-cells. And finally, CD62L or L-selectin, which is expressed mainly in naive and central memory T-cells. Last but not least, we have left the FITC channel free for drop-ins. So the user may add complementary antibodies for the identification of more specific T-cell subpopulation or include FITC conjugated reagent for the detection, for example, of CAR T cell or other antigen-specific T-cells. So let's jump now into the main T-cell subpopulation that can be identified with these combinations. We will see first the different CD4 positive T-cell using the maturation marker. I mean CD27, CD45RO, and CD62L. So we will use mainly two dot plots, the CD27 versus CD45RO and the CD45RO versus CD62L. Let's start with the naive CD4 cell. In dark green here, these cells are characterized by high expression of CD27, but dim or negative expression of CD45RO. And these cells have also a high expression of CD62L marker. Then along the maturation pathway, we can identify the central memory T cell. This cell has a high or a positive expression of CD27 and CD45RO, but also they present a high expression of CD62L marker. In the case of a transitional memory T cell, this cell expresses a slightly lower level of CD27 than central memory T cells, but this transitional memory T cell still shows high expression of CD45RO, but lose expression of CD62L. And what happened with the effector memory T cells? We see on the plot that they are negative for CD27 and they but positive for CD45RO, and they present variable expression of CD62L. And finally, the terminally differentiated effector cells. These cells are CD27 negative and CD45 dim or negative, and they show variable expression of CD62L marker. So using now the same two dot plots, we see how looks the pattern of the CD4 positive cells from naive to the terminally differentiated effector cell. In the case of CD8 positive T cells, a similar subpopulation can be identified in terms of immunophenotyping using the same two dot plot you can see here with the corresponding immunophenotype. However, an additional subpopulation can be found among the CD8 positive cell that is not easily found in the case of CD4 positive T cell. Those are the early effector CD8 positive T cell and they are characterized by dim or positive expression of CD27, a negative expression of CD45RO, and also negative expression of CD62L marker. So, but how we can take a look over this T-cell pathway? So let me just show in this slide some multiparametric advanced analysis tools that are included in the InfiniSight software that are particularly useful when you are analyzing sample stain with this T-cell subset panel. So in this slide, we are presenting data coming from 3 patient files at the same time. So because we are using the file merge tool that we have in InfiniSight software, in InfiniSight, this merging is meaning that you can overlay the file. So the resulting file of this merge process will contain all the events in the same view. So we can focus first in this APS diagram. This automatic population separator separate the event of the sample, taking into account all the parameters of your file, and separate the population based on the likelihood. So the events that are similar will be close together. The events that are farther away is because they are immunophenotypically less similar. Let's see, for example, here in dark green, the naive Tcells, and in the other part of the diagram, in light green, the terminal differentiated effector Tcells. In addition, on the right part, we can see the maturation diagram. So using the information coming from several files, we perfectly see how the expression of the marker evolve along the different stage of the maturation. So let's see, for example, over the CD8 diagram, and we just saw the CD27 here in green expression start with high expression in naive state, but finally lose expression in the last state. And the same happened with the CD62L marker. The expression of this marker has a variable expression in the effector stages. So you could create a maturation database with a normal sample, enable a quick overview of your abnormal sample comparing with this maturation diagram. And all these tools are included in the InfiniSight software. But what is the aim of this panel? So what can be the potential uses for this combination? Well, firstly, monitoring oncohematologic patients, taking into account that memory T-cell subpopulation can change during the response to the tumor. Another important aspect is measure the response to vaccine or infection. In this context, the FITC channel can be used for the identification of virus-specific T-cell population. And also, in a similar manner, the free FITC channel could be used for the separation of CAR T-cell from the endogenous T-cells, and this could be one of the most important intended use of this kit. So now, during the presentation, we will take a look at some example of memory T-cell studies that we have found available online for each of these applications. So let's see the first application to monitor oncologic patients. In this first example, the authors analyze the distribution and function of the different memory CD4 and CD8 positive T-cell subset in patients with non-small cell lung carcinoma compared with healthy donors. They use, in this case, peripheral blood and lymph node samples. And in this case, they use CCR7, that in terms of immunophenotyping is similar to CD27. And they use, in this case, CD45RA, that is a marker of non-memory cell instead of the CD45RO that is expressed in memory T cell. So the analysis of the CD4 positive T cells subset, in this case, in this disease, indicate that the peripheral blood and lymph nodes contain similar frequency of the different cell subset population, both in healthy donor and in patient. However, for the CD8 positive cell, the terminally effector cell, you can see here in dark, represent between the 50 and 70% of the total CD8 positive T cells. And in contrast, the frequency of the CD8 positive effector memory T cells was lower in the blood of this patient compared with healthy donor. So in this point, it's relevant to say that although the analysis of T cell memory immunophenotype is not part of the diagnostic or prognostic process in oncology, it can be implemented easily to the clinical routine as a simple and fast method to monitor the immune response to the tumor or to the treatment. The second example I want to show, this is another example of applicability of this T-cell panel. We will review here a study of infection with a worm. Here the authors wanted to compare the frequency of CD4 positive and CD8 positive T cells in healthy donor and in patient infected with a worm. You can see here on this diagram that they could follow the same strategy for getting T cell. So they can use the CD45 and then CD4 and CD8 to classify the helper and cytotoxic, and then use the marker of TCR7 in this case and CD45RO to classify the different subsets of T cell from naive to effector T cell. In this case, regarding the CD4 positive T cells, they observed that after the treatment, the people infected with the worm has a decrease in naive T cell, and they present high level of central memory T cell, but also they recover basal levels of effector cells. So with this antibody combination, you can perfectly follow up what is happening with the T cell response of the patient during the infection and after the treatment. Third, here we saw another example of the importance of the study of the T cell subset. In this specific publication, the response to COVID-19 is analyzed. So the author in this case studied the overall and the SARS-CoV-2 specific memory T cells in patients who have recovered from COVID-19. And how they did. The peptide pentamer were synthesized comprising peptide coming from virus-specific T cell. So they conjugate this peptide with p-fluorochrome. Remember that in this case, the p channel free helps to identify virus-specific T cell, and in our solution we have left the FITC channel free, but it is the same approach. So with this strategy, the authors can compare the memory phenotype of COVID-specific T cell with the overall circulating T cell. So you can perfectly differentiate the overall T cells and the T cells that are responding specifically to the COVID-19 virus. So regarding this issue, it's important to comment that the T cell immunity is getting more relevant in monitoring the COVID-19 general immune status, and also in analyzing the response to vaccination. It has been mentioned before, another exciting application for this panel is the immune monitoring. And the most common example could be the CAR T-cells, because it's well known that the CAR T-cell therapies has experienced aggressive in recent years as the emerging therapies for the treatment of both hematologic and solid tumor. So the free FITC channel here allows the addition of reagent for the identification of CAR T-cell, and we can follow different strategies. We present here two different strategies. Firstly, we can include an antibody that recognizes one of the structural domain of the CAR. You can see here, we can approach different antibodies that recognize different parts of the structure of the CAR. But the second strategy, and more interesting, the use of the target protein of the CAR. And the target protein of the CAR labeled with the FITC fluorochrome. In this case, the CD19 is the target protein of the CAR. So the advantage of this method is that the labeled protein can bind to all CAR that are directed to CD19 independently of the structure of the CAR. But also, doesn't matter the strategy you follow, you can follow. The advantage of this panel is that you can monitor the immunotherapy. Interestingly, in the last few years, it has been proved that the CAR-T products that are rich in central memory T-cell are more long-lasting and more effective in the tumor clearance. So we could follow the previous strategy to select the specific memory T-cell among the CAR T-cell, and then expand ex vivo specifically the population more effective in the treatment. And this fact had like the relevance of our combination in characterization of T-cell product when we are generating CAR T-cells. In this part of in the lower part of this slide, we can see an example of monitoring the infused CAR T-cell in time. So you can perfectly follow up when the treatment is more effective, and also what is happening days after the treatment. And we can see if we still have effective CAR T-cell or what is happening in this patient with the CAR T-cell previously infused. So the concluding remarks of today is that we offer a single tube 7-color panel that is commercially available with this reference. This solution guarantees a simple application and more reproducible results. The panel is easily adaptable to the different studies, as the free FITC channel allows the addition of FITC label antibodies or FITC label proteins. Furthermore, the staining and acquisition of the sample is fast, taking less than 1 hour in this case. And finally, our carefully selection of antibodies and fluorochrome is going to facilitate the accurate immunophenotype of the different subset, naive memory T cells, and is going to allow the follow-up of all the T cell subpopulation. And this characteristic make our solution valid for all these 3 possible applications: monitor the immune response to the tumoral therapies, monitor some immunotherapies like the CAR T-cell, or even evaluate the response to vaccine or other types of infection. So finally, thank you again, Biospecific, for helping me. Thank you, all of us, for listening to me today. If you have chance, don't forget to check our website and contact us directly at this for more questions, or if you are interested to know more. Thank you for your time.